Hi everyone, welcome back to Useful Genetics. This is the start of Module 2, the overview video, where I'm going to give you a bit of a flavor and perspective on what's coming up in this module. The module's about mutations, so you might think it was going to be pretty dry. I mean, how interesting can changes in molecules be? But in fact, mutations cut to everything that we care about. We talk about harms and benefits, stability and variation, research importance and personal significance, topics such as cancer. We'll talk about immediate changes and evolutionary changes, the kinds of mistakes that molecular machines make, and we'll be tying together physical changes and informational changes. Informational changes really come to the fore in thinking about mutations. So even though we're talking about changes to molecules, almost all of the language that we're going to be using is the language of information, copying, proofreading, correcting, mistakes, recognition. Now, to give you a bit of perspective on the lectures, I've grouped them. So the first two lectures are about the basics of mutations, in particular what they are, how often they happen, and why they happen so rarely, because in fact the rates are astonishingly low. We'll talk about the consequences of mutations in a series of lectures, why most mutations are harmless, and the kinds of harm that other mutations do. We'll consider briefly beneficial mutations as well. We'll make the important distinction between somatic mutations and germline mutations. The latter we can pass on to our offspring, but the former, which are restricted to cells that don't pass to the next generation, these are the mutations that cause cancer. We'll then spend a couple of lectures talking about mutagens, things that increase the mutation rate, and whether or not these are things we need to worry about. And then finally, we'll pull things together into this big evolutionary context, thinking about how natural selection acts on mutations, how genes evolve, and how we can use the accumulation of sequence differences to investigate how evolution happens. Now, coming up next is the first lecture on mutation basics. Here's DNA polymerase feeling good because it made a mistake and it corrected it. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is the start of Module 2 where we're talking about mutations. And in this first lecture, we're going to talk specifically about errors in DNA replication. We'll first introduce why we care so much about mutations, and then we'll talk about how mutations arise and about the events that can happen that determine whether a mistake by DNA polymerase actually turns into a mutation. So the basic definition is that a mutation is a change in DNA sequence. And we can clarify that by describing some constraints on this. First, the DNA sequence has to have become different than the normal standard functional DNA sequence for that organism, usually different than the sequence that was there before. If we consider it as if it was text, because this is very much an informational concept, we could say that, for instance, if the word tire was the normal word, the normal sequence, then any of these changes of replacing the I with a Y, which makes a word that has the same meaning, replacing it with an O, makes a word that has a different meaning, replacing it with an X, well, that's not really a word at all, but it's still a string of text. Which brings me to the second point, that the change must produce something that's still structurally normal DNA. So this is structurally normal text because it's just got the normal letters. But these are not structurally normal text. Question marks don't belong in the middle of the words, nor do snowflakes. This is a broken character, equivalent to a defective piece of type, and a symbol from another language. So none of these are mutations. And part of the reason that they're not considered mutations is that a mutation must be inherited by daughter DNA molecules. If you have to be able to pass it on. And these structures are not things that DNA polymerase would recognize, and so they can't be inherited. A mutant is the other related term. A mutant is an individual that has a mutation. So the mutation is the change. The mutant is the individual or the cell with the change. Now, there's lots of reasons to care about mutations. Mutations reduce fitness. They cause harm to individuals and they cause extinction of species. They cause cancer. We'll talk a lot about cancer and how mutations are the cause of cancer in Module 4. Mutations make us worry a lot. We'd like to understand mutations better so we know what to worry about. On the other hand, in an evolutionary framework, mutations generate the heritable variation that's the raw material of evolution. If we didn't have mutations, there'd be no natural selection, and evolution wouldn't have happened. Finally, accumulated mutations tell us how evolution happens. They provide a kind of historical record, and we'll talk about this in a later lecture. Now, here's a diagram showing how mutations arise in DNA replication. So here's DNA polymerase in the process of replicating a template strand, making a new strand. And it should put in a C at this position opposite the G, but instead it puts in a T. It makes a mistake. And this creates a structure that we call a mismatch. However, a mismatch is not a mutation by the definition that we just went over. Now, when this DNA is replicated again at the next cell division, each of its two strands serves as a template for the synthesis of a new strand. Now I've colored the new strands green. And what you see is that one of these double-stranded DNAs, this one, contains a changed base pair. This is a mutation. The other daughter DNA molecule, double-stranded DNA, contains normal sequence DNA. Now, most mutations start out as mismatches, but most mismatches don't become mutations. In fact, less than 1% of mismatches become the kind of mutations that I just showed you. And that's because of a function carried by DNA polymerase called proofreading. Proofreading is a term, of course, from English publications, from text, and it means in the context of DNA replication, it means exactly the same thing that it means in the context of checking the quality of type of written material. So here's our DNA polymerase, and it's made a mistake. It's created a mismatch. Now, in the previous slide, I showed DNA polymerase just chugging on ahead. But in fact, DNA polymerase doesn't do that. Most of the time, when DNA polymerase puts in a base, it sort of feels behind it and checks to see how well that base fits. And if that base doesn't form a proper base pair with the template strand, DNA polymerase backs up, and this brings into position a part of the DNA that's able to cut out the defective, the mismatched base, which then allows DNA polymerase to proceed synthesizing, inserting a correct base, and then continuing with the synthesis of the DNA. So in the absence of proofreading, the error rate is about 10 to the minus 5 to 10 to the minus 6. So that's mismatches per base pair. That's about 1 in 10,000 to 1 in 100,000 base pairs is the wrong base. Now that's actually an extraordinarily accurate replication rate for a molecule to be able to accomplish, but it's not good enough. With proofreading, the error rate goes down to approximately 10 to the minus 8th, which is an astonishingly high fidelity rate. But it's still not good enough because our genome is so big. And the cell solves that by bringing in a completely new independent repair system on top of proofreading, and that repair system is called mismatch repair. It does exactly what its name suggests it would do. So here's our DNA molecule containing a mismatch. This time DNA polymerase didn't notice it, didn't proofread it and correct it. It's been left behind. Now, so far I've drawn DNA with mismatches as if it was structurally normal, but in fact, the inability to form a base pair here creates a deformation in the backbone of the DNA. I'm just going to draw it as a little kind of bump. And mismatch repair enzymes travel along the DNA, feeling the shape of the DNA. And when they encounter a place where the shape of the DNA is wrong, they initiate a repair process. And what they do is they cut out the damaged section, the wrong base, and some bases around it. And then they leave the DNA. This creates a gap in the DNA where a repair polymerase, a special DNA polymerase called a repair polymerase, can come in and fill in the gap using the template strand to insert the correct base. And then the DNA repair polymerase leaves the DNA, and we've restored functional DNA. So the efficacy of mismatch repair is such that it corrects about 90% of what proofreading overlooks. So the final error rate is approximately 10 to the minus 9th errors per base pair, which is an extraordinary fidelity for a molecule or a molecular complex to achieve, far better than we can do ourselves. So here's a question. If DNA polymerase inserts a wrong base that isn't corrected, it isn't corrected by proofreading, it isn't corrected by mismatch repair, and then the DNA gets replicated again when the cell next divides, can the mistake still be corrected? And the answer is no, the mistake can't be corrected. Because the changed DNA is now structurally normal, there's no process in the cell that can detect that it's changed from what it used to be. It's now a mutation. And to help you, it might help you to think about that, to remember the image that we looked at in a previous slide, where we have, after the second round of DNA replication, we have one DNA double helix where we have correct base pairing, but the base has changed, and we have the other double helix where the base is still normal. So, what have we done? We talked about why we care about mutations, and then we talked about how mutations arise mainly because of mistakes made by DNA polymerase. We'll also talk about later about how mutations can arise due to the creation of damaged DNA, DNA that's not structurally normal. So when DNA polymerase creates mutations, the first step is that the polymerase puts the wrong base pair in when it's replicating the DNA. That, however, is followed normally by two correction steps. First, a proofreading step, and then a mismatch repair step, which between them reduce the rate of mistakes in DNA replication by more than a factor of 1,000. Coming up next, we're going to talk more about mutation rates, and we're going to talk about how natural genetic variation is the product of mutation rates and the action of natural selection on the mutations that are created. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 2B, and we're going to build on our discussion of DNA replication errors in Lecture 2A to talk about mutation rates, how often mutations arise and why, and about the consequences of mutations, and introduce ideas about their evolutionary history. As we said in the previous lecture, new mutations arise rarely, in part because DNA polymerase is very accurate. So we'll start with we're going to extrapolate and think about mutation rates before we were thinking about error rates in DNA replication. So here's a number. In humans, from parents to child, there are about 1 to 2 times 10 to the 8th new mutations. But what I've done here is I've left off the units. So what do you think the units are here? 1 to 2 times 10 to the minus 8th new mutations per what? Per genome, per gene, per base pair? In fact, it's new mutations per base pair. This is 10 to the minus 8th is a very small number. We have many, many base pairs in our DNA. But this isn't a complete thinking of per what. This is per amount of DNA, but we also need to ask per what sort of time unit? Per year, per generation, per cell division, which is effectively a DNA replication? And the answer is it's 1 to 2 times 10 to the 8th new mutations per base pair per generation. Now, we can reconcile this with the numbers that we talked about in the previous lecture, in that we said there are about 10 to the minus 9th remaining errors after mismatch repair and DNA proofreading have done their best to eliminate the mistakes that DNA polymerase makes. So there's about 10 to the minus 9th errors per base pair per cell division, we'll say, which is the same as a DNA replication, cell division. That's a smaller number than this number. And the reason is we're now considering not a single cell division, but all the cell divisions between the fusion of eggs and sperm and the production of the next generation's egg or sperm by the adult. And that's a fairly large number of cell divisions, more than 20 for sure. Now, new mutations arise very rarely, we just said. In fact, very, very, very rarely. A very accurate process, provided the DNA that DNA polymerase is using is not damaged, as long as it's structurally normal DNA. And this is accomplished by a combination of proofreading by DNA polymerase and mismatch repair. But DNA also can get damaged, and we'll talk more about this later. Cells have evolved ways to repair DNA damage because otherwise this DNA damage would cause mutations. And the two processes, which we're not going to talk about at all, I just want to give you the names of them, are called excision repair, which cuts out damage, and recombination repair, which patches damage. Now, how many new mutations do we get? We said 1 to 2 times 10 to the minus 8th per base pair per generation. What does that mean practically? We don't think in terms of base pairs. That's about somewhere around 100 new mutations per diploid genome. And we can do the arithmetic to see that that is about what we get. That means that on average, each new baby has approximately one, sometimes none, sometimes one, sometimes two new mutations that were not present in their parents, that changes an amino acid in a protein. So this means that we're constantly generating new genetic differences. Now, one more piece of information, which I'm just going to raise it here and come back to it in lecture 2H, and that's that most of these new point mutations arise in the father, in the production of gametes by the father, not in the production of gametes by the mother. And we'll talk about why later. Now let's think about the consequences of these mutations. And the most important point to know about mutations is that most mutations have consequences that are either neutral or harmful. Mutations that are beneficial are very rare. So why is this the case? Well, first, most mutations are neutral because they occur in places in the genome where they don't make any difference to gene function. We'll talk quite a bit more about this in lecture 2C. Most of the rest are harmful. Very few new mutations are beneficial. And that's because living systems, the genetic DNA sequences that specify all the properties of living systems, are very well adapted. They're the product of billions of years of natural selection for better and better function. And that means that if you make any random change, and that's what a mutation is, it's a random change, the odds that this change makes the system function better are very, very small. In the same way that a random little change to the innards of your computer is very, very unlikely to make it function better. So we can take this understanding of the functional consequences of mutations and apply that to thinking about the genetic differences that we see. Fundamentally, all of the genetic differences that we see, all of the differences, for instance, that we talked about in Module 1, they all started out as new mutations with the same spectrum of neutral, harmful, and beneficial as I just described. Natural selection then acts as a filter. It eliminates the bulk of new mutations that are harmful, and it allows to persist all the new mutations that are neutral and increases the frequency of any new mutations that are substantially beneficial. Those new mutations, the beneficial ones, are a very small fraction. Almost all of what we see as natural genetic variation is neutral mutations that have accumulated over evolutionary time. So we've talked about the rarity of new mutations. We did some arithmetic. And remember that they're extremely rare in the context of per base pair. But when we extend that to think about whole genome, our genomes are so large that we inherit new mutations with every generation. Almost all of these new mutations are neutral or harmful. The neutral ones tend to persist. The harmful ones tend to be eliminated by natural selection. More slowly if they're masked by the presence of a good copy of a gene, which is something we'll talk about in Module 3. Neutral mutations in particular accumulate over evolutionary time, and it's these neutral mutations that are largely responsible for the many DNA sequence differences that we find between different people. And then a small subset of these neutral mutations that cause phenotypic differences that we see are responsible for the phenotypic differences that we see between different people. Coming up next, we're going to think in more detail about why most mutations are harmless. I hope to see you there.